So, well, firstly, thank you very much to Yves for giving me again the opportunity to come here and talk a little bit about the work that we're doing in the department. So I've slightly changed my title. I've put a short evolutionary viewpoint, and perhaps like Thomas said in the previous talk, he says he's not an expert in electromagnetic radiation. Well, I'm no evolutionary biologist, I'm a materials engineer. But I came across, or together with Mason, who you'll be seeing later on today, some interesting observations which might give at least an evolutionary touch to this symposium that I'd like to share with you. So the first thing I'm going to do is to discuss a little bit about these ideas. And then what I'd like to do is to give you an example of where architecture is found in biology, so where you have an architectural control parameter which is used to get different functions. And then I'd like to talk at the end a little bit about processing. So what do we know about how natural materials are being processed? Okay, so, well, some pretty pictures, but biomaterials are of course everywhere. And if we look around, well, wood contains, if you think of it as a structural material, it's containing cellulose, hemicelluloses, and lignin. Cuticles, of course, from arthropods, they contain chitin. Sometimes they contain mineral, and there's a variety of other proteins which they also contain. Shells contain calcite, aragonite, so mineral particles, also together with chitin and protein. Fungal cell walls contain chitin, bone contains collagen, mineral, a variety of proteins, and of course water plays an important role in all this. So when we talk about biological materials, in many respects you can think of them a bit as being composites. But what's interesting about biological structural materials is that they're, if you look at it with a very broad brush, there seems to only be a very limited number of building blocks that are used in structural materials in biology. And I say that with a very broad brush, of course, because if we take collagen, for example, there's not just one type of collagen, there's many different forms of collagen, but the rough structure, one could say the structural motif at least, is fairly well conserved. Silica, calcite, hydroxyapatite, cellulose and chitin. I mean, apart from that, well, there's quite a lot of mineral phases which are found in biology, but there's a fairly limited list of building blocks or materials which are used to construct structural materials in biology. Of course, proteins play a very, very important role here, and Peter highlighted that a little bit yesterday in his talk, where by introducing very small amounts of proteins, for example, in glass sponges, for example, you can improve properties drastically. So I'm not saying that proteins are not important. It's just quite a curious observation that, first of all, there's a very few, the major components of biological materials are fairly limited. So, of course, I can't resist showing a material property chart. So this data also comes from the CES database and shows a range of strength and Young's modulus data for a variety of different natural materials. And what do we get here? You notice that there's not many holes in this space here. So despite the limited number of building blocks, you can fill a very large range of, or area in this material space. Okay, I chose strength and Young's modulus. Of course, if you look at other properties, maybe this is no longer the case. And what does this mean? It means the diversity in the materials properties comes through the material architecture, so the way in which these components are put together. And what do we mean by architecture? Well, very quickly, just again, you've seen this before. For example, in bone, we can talk about a hierarchical architecture or a multi-level architecture. We're going from the macroscopic scale, there's a pointer here, macroscopic scale, and it's a little bit out of focus. Well, let's see. Oh, we've got a nice little arrow there. Down through the different length scales, you can see different levels of organisation where we have lamella-like organisation of collagen, we have fibres which are then mineralised, and then there's organisation at the nanometer scale. And that's better. Okay, so someone that knows about electromagnetic radiation. Okay, so what does this mean? Through this hierarchical architecturing, you can take a mixture of, say, collagenous-like proteins, mix them together with mineral particles, and then you can end up with composite materials which combine the toughness, for example, this isn't a toughness versus stiffness plot, the toughness of collagenous materials with the stiffness of minerals, and giving you high performance materials. So, okay, so this is just a quick example of how at least architecturing can give rise to new properties. So I came across, a couple of years ago, a very interesting paper by a guy called Andy Knoll from Harvard, looking at the evolutionary history of biomineralization. And what he did was he took the eukaryotic tree of life and started placing on there in the different groups. So you can see we're up around here somewhere. You've got mollusks, you've got arthropods, fungi, plants, etc. Started placing on this chart where you can find different biominerals. And this is quite interesting. So if we look at calcium carbonate, for example, you can see calcium carbonate can be found amongst animals. There's plants that produce it. There's also sort of spread around the entire eukaryotic tree. He also looked at silica, again, fairly broadly spread, and also looked at calcium phosphate. So something I was sort of amused myself in the last weeks or so was to start saying, okay, well, what else can we plot on this graph here? We can plot cellulose, and cellulose can be found, of course, in plants, that's obvious, but there's slime molds that seem to be able to produce cellulose. Fungi, there's some thoughts that some fungi at least can produce cellulose, although it's somewhat debatable. Interestingly, there are also animals that can produce cellulose, so the tunicates. Somehow thought that this is lateral gene transfer. They borrowed the ability to do that from bacteria. But again, you see this is quite widely spread. If you look at chitin, again, widely spread through the animal kingdom, collagen as well. Now, one big caveat on this is that this is an approach of a materials engineer filling in this plot here. I've discussed with a few people talking about how to fill in this plot. So it's certainly not exhaustive, and a lot of work needs to be done here to make, say, an exhaustive list through all taxa about whether different materials have been found there or not. But already with this plot, it gives quite an interesting sort of consequence, I think. And the first thing is that these building blocks that I'm talking about, these component materials, are spread across many taxa. So that's the first thing. And what's interesting is if you look in the literature and see when these different groups appeared, it's quite likely that these building blocks appeared before the Cambrian explosion. So what the consequence of this is that over about 500 million years, the building blocks haven't changed. And that may give rise to the question, over the last 500 million years, when we've had so much change in structure and form in the life that's on Earth, has evolution just operated on the materials architecture? And that's where I come back to the materials architecture, the topic that is the focus of this symposium here. So this is very preliminary, perhaps quite, probably there's quite a lot of errors in this, but it's an interesting question that's worth pursuing. So how would one pursue such a question? Well, one way of doing this is to do, say, a systematic comparison between different taxa. And you're going to see talks later on today by Mason and Ron, where they'll be talking about the architecture and structures that you find in sharks and rays, as well as fish and fish bone. Vanessa is going to be talking about architectures that you find in a group of plant species. And basically, what's interesting about this work is they're really comparing in a almost systematic way, or in a very carefully chosen way, differences in material architecture between different species, between different groups. Another important way of looking at this is to say, well, we need to understand how architecture controls function in different systems. And Garrett will be talking a little bit about arthropod cuticle later today, I believe. Vanessa and Sebastian will be talking about how architecture may control movement. And the last point that I would I'll be talking in the next part of the talk a little bit about this. And the next part that one could use is to try and understand how architecture is formed in different systems. Okay. So now I'm going to take a step aside, and I'm going to talk a little bit about one example, and that's the example of plants, where architecture can control function. So let's look at a piece of wood. Well, here's a block of wood that's been cut out. If you zoom in on a small 100 micron by 100 micron block of that, it looks a little bit like this. You can see the growth rings. You can see variations in cell size. If we zoom a little bit further, now we have to zoom in more in terms of imagination, perhaps. And we have a schematic here of the plant cell wall, or the cell wall in wood. And in this region here on the outside, we have the primary cell wall. So this is cellulose microfibrils embedded in a matrix of other polysaccharides. And the major component of the wood cell wall is this thick layer here, the secondary cell wall layer. And this consists of, again, cellulose fibrils. Peter mentioned it. These are stuck together with a hemicellulose matrix, which gives interesting properties in terms of plasticity. So these things can plastically deform. And if you look, say, in a cross-section in the SEM, in some special cases, you can also see the consequences during drying of this microfibril angle, where you can see, if you cut carefully, this splitting that's occurring here during the drying in the SEM, which gives you an indication of the microfibril angle. Another way of measuring it, of course, is to use X-ray diffraction, methods that I'll show you a little bit later. So there's one important design parameter here that plants can use, and that is the angle at which this cellulose microfibril is wound around the cylinder. And this is one of the things that Olivier Boisiz was talking about yesterday in some of his experiments, to try and see if he could mimic these sorts of structures, and then to see the effect out of metals, and to see if he could see the effect on the mechanical properties, because you see quite an effect on mechanical properties, at least in plants. And Damien Fabrègue was also using his plasma sintering method to see if he could produce also similar structures. And it'll be very, very interesting to see whether you get improvements or consequences in mechanical properties in the results. So what do you see in plants? So just schematically, if your microfibril angle is close to 0 degrees, so if you imagine all your fibers are oriented along the tube here, of course, then if you think about composite theory, then your cellulose fibrils are very stiff. They're oriented along the axis, and so you'd expect if you pull on something like this, you're going to have a very stiff composite material. If your fibers are now oriented at an angle and wound around the cell, you can imagine that the matrix in between the fibers is going to be more loaded. And that's going to have the consequence, of course, it's going to be softer. And if you go to larger and larger angles, you would expect to have even more flexible and soft materials. So this can be measured. So here's a collection of data that was made by Michaela Eder, who unfortunately can't be here today, of the tensile stiffness versus microfibril angle of a variety of different wood fibers. So in the white circles, these are macroscopic wood blocks. So these contain many, many individual fibers or cells of wood. But what's also plotted here are individual wood cells. And you see that there's a very clear trend here that going from, if you have high microfibril angles, so if we do the arm waving thing again, so angles like this compared to the longitudinal axis of the cell, you'll have low stiffnesses and high stiffnesses for high microfibril angles. So whereabouts can we find these different microfibril angles? So let's go for a walk in the woods in Potsdam. For those of you that were here earlier on in the year, we're going to go for a walk again in the woods of Potsdam. And we're going to have a look around and see whereabouts we can find examples of these different microfibril angles. So if we look at, say, a young tree, what we typically find are quite large microfibril angles or intermediate microfibril angles. Now, why is that? So if you can do this the next time you go out into the forest, if you take a tree like this and bend it to the ground and let go, it springs back up again. It's quite resistant to mechanical loads. However, if you now take a large tree that's now had time to grow, if you try and bend that, of course you don't see much motion, but more importantly, it's loaded very heavily under its own weight. So the mechanical requirements of the tree have changed drastically during its lifetime. And as a consequence, you find smaller microfibril angles, so stiffer material being produced in the older, or so the newer wood, or in older specimens. And this is quite interesting because this is one control parameter that the plant can use. This is just changing angle, and you can get quite a large change in mechanical properties. In addition, there's also specialized tissues in plants. For example, there's compression wood, which is found on the underside of certain species. And this has, interestingly, a very large microfibril angle, and we'll come to that in a minute. This allows one to develop large compressive stresses underneath the branch. In other species, there are specialized tissues which are able to create high tensile stresses on the upper side of a branch, and this does this through creating very low microfibril angles. And we won't go into that example. That's the example of tension wood, but this enables tensile stresses to be created, so that as a branch grows and it gets heavier and heavier and heavier, there's reaction stresses that can form in a tree to pull the branch back upright again. And so if we take normal wood, for example, so this is the standard wood that you find, say, in the trunk of a tree or whatever. If you have a microfibril angle of about 10 or 15 degrees, this can produce small amounts of tensile stresses. The compression wood, which sits under the underneath, can produce compressive stresses, and then the tension wood, which we'll talk about a little bit later. So this is an experiment that was done in the group of Ingo Birgit, which is quite nice to show. If you take some normal wood and compression wood and glue them together into a, see if I can get this thing to work. Okay, it's starting to work. If you glue them together, what you observe is bending, or you put them into a humidity chamber, you change the humidity, the water tends to go into the matrix, so the material surrounding the fibers. You end up with bending, of course, of this bilayer. And how does that occur? Well, we can do this thought experiment. So we take our cylindrical cells, or assume they're cylindrical, with two different microfibril angles, so 50 degrees and 20 degrees, for example. If we make a cut down one of the walls of both of these cells, we open them up, and you see we have our flat piece of tissue here, a flat piece of tissue here, and in green I've highlighted schematically the orientation of the cellulose, the stiff cellulose microfibrils. In light green is the hemicellulose. So this is the material which will absorb water and swell. So we can cut again a small piece out of here, so that we've got now a rectangle where the diagonal is now made of one of these microfibrils, and again another one here. Now, if the green material swells, typically it's quite soft, so it can't change, to a large degree at least, the length of this red line here on the diagonal. Of course, if you take a rectangle and you try and maximize the area during swelling, the consequence of that is to, of course, try and tend towards a square. So if we do that for, and we'll have to go backwards and forwards for the top one and the bottom one, if we do it for the bottom one and don't change the length of the red line, we see we get an extension. Oops. If we do it for the bottom example of the low microfibril angle, we get a retraction. So just by changing angle, you can go from tensile stress to compressive stress. And that's just one architectural parameter that plant tissues can use. So I'm going to give one example of work of Rivke Elbaum, who was working in the department on a Humboldt fellowship, and she's now at the same institute where Ron Shachar comes from, at the Hebrew University of Jerusalem. And what she was interested in was the awn of wheat. So she was looking at wild varieties of wheat. So this is a typical thing that you'll find, or you would find at, say, early autumn out in the fields. So in modern varieties of wheat, the seeds stick onto the wheat plant. In old varieties of wheat, it looks quite different. These seeds will fall to the ground. Of course, old varieties of wheat were not evolved to be to have their seeds spread by humans. If you look on these, and it has these enormous antenna-like structures called awns. If you look at this in closer detail in SEM, what you notice is on these awns is these spikes, which are all oriented in one direction. These spikes, interestingly, are made out of silica. So an example of a plant system that can produce a biomineral. And if you make a cut through the cross-section and look at this using scanning acoustic microscopy, which gives you an idea of the mechanical properties, you see that there is a region of high intensity, so high acoustic impedance, and also reflectivity, I think, and one region of low intensity. And by using small angle X-ray scattering, wide angle X-ray scattering, what they managed to observe is that there is a strong orientation of the cellulose microfibrils in this cap region, so on one side of these awns, and a more random orientation in the other region. So what are the consequences of that? So that means that on this side, if you have any changes in humidity, there's going to be no change along the longitudinal direction of the awns. And in the lower region, if you imagine you get swelling, you're going to get isotropic swelling in all directions. And this has the consequence that if you now change humidity, these awns bend. And the idea that Rivka and Peter came up with was that these little hooks that are on there enable the awn to dig itself into the ground. It can only move in one direction, and it swims along the ground to propel the seed into or away from the mother plant. This is an example of microfibril angle being controlled on two sides of an organ. It's essentially the same chemistry on either side, but it's this architectural parameter that's being used to come up with a new function. There's another example here. It's the work of Matt Harrington, It's the example of the ice plant. And what happens here is, so this is a photograph taken up on the northwest coast of Western Australia, well, close to where I come from. But you can find these plants pretty much in coastal regions, in arid regions around the world. If you have changes in humidity, this seed capsule here, which appears after flowering, changes in humidity, you see no changes at all. However, when it rains, when enough water is around for germination, so you're placing a drop of water on this seed capsule, you see opening of the seed capsule. The seeds are now available to be released into the environment at a time when there's sufficient water for seed release. Again, the only living things in here are the seeds themselves. Everything around, like in the wheat awn in fact, is dead tissue. So this means that what's controlling the opening process is due to the architectural properties of the material itself. You can isolate the motor which causes this opening, this keel here. If we observe this as a piece of water or as it's placed in water, you see this opening process. And how does this work? Well, this doesn't operate through cellulose microfibril angle. It operates perhaps in a nice contrast to the talk previously through the use of clever designs of honeycombs. So in the dry state, well, you can just make out the individual cell walls. In the wet state, you see you have this diamond honeycomb structure. So basically you have a honeycomb which is compressed in one direction. If you now, I've lost my mouse again. There we go. If you allow the material inside the honeycomb to swell, you'll get very anisotropic elongation, much more elongation that you can get compared to playing with microfibril angle. But again, the material is essentially cellulose and a few other polysaccharides. So what we've seen here in these few examples is how in plant tissues at least, by controlling very simple architectural parameters, one can control function and can achieve quite a wide range of functionalities. So if architecture controls function, then there's a question, of course, related to processing. And this was a theme, of course, yesterday in many talks. Because in nature, of course, you see absolutely sort of wonderful structures which play a very important role, of course, in function. So in bone, you see this wonderful liquid crystal-like structure. I think Garrett will be talking a little bit about similar sorts of structures that can be found in stomatopoods. Of course, we have wood here. So how are these sorts of structures formed, and what is known about this? So I'll talk very briefly about plants, and then I'm going to switch kingdoms and talk more about animal cells. But in plants, at least, a lot is known about the machinery, and that's mainly related to the work of Cosgrove. And what they've managed to show is that the cell cytoskeleton, so inside the cells, the microtubules, they can control the deposition of cellulose. However, up till now, what is not understood is how in a tree, for example, in one region of tissue, a particular microfibril angle is chosen during deposition, and in another region of the tree, at the same time, another microfibril angle is chosen during growth. It's known there are properties such as gravitational sensing, mechanosensing in many living systems. So these things are probably responsible, but really the link about how the architecture is controlled is something that requires a lot of active research. So I now move back to these sorts of structures, and there's actually, it's related to a question that was in the audience, I think yesterday, it turns out that there are a lot of biological processes which could be described by liquid crystal ordering. And this is the work of Professor Georgi, who I think is also at the Collège de France, is just up there. And in this very, very elegant work, what they managed to show is that collagen self-organizes, please correct me if I'm wrong, collagen manages to self-organize on its own into very similar structures to those that you observe in bone. Similar things have also been observed for cellulose and chitin. So essentially, if you have very long polymers, there is a stabilization of these liquid crystal phases, which can give a hint at least to organization of these tissues. So self-organization can indeed lead to complex architectures. So in some of the work that we're interested in our department is to try and look at what is the role of cells on this self-organization behavior. So first of all, I'll just quickly describe the experimental system. We use rapid prototyping to make or additive manufacturing to make controlled shapes in a scaffold. We then seed cells on them. We're using osteoblasts, so bone-like cells, but we've also played with fibroblasts, and together with collaboration partners, we've been looking at primary cells. We then observe how the tissue's formed in there, and typically what you observe is the tissue starts forming like this inside the pores, and when we'll look through the pores in cross-section using light microscopy, and we can observe how the tissue is forming as a function of time. So the sorts of experiments that we get, so this is examples of circular pores. You see that this brown material is slowly growing with time, so the hole is getting closed and occluded with time. In the corners of the pores, you see also material being deposited mainly on the corners and not so much on the flat surfaces of pores. Turns out one way of describing this is by describing the tissue physically, or as a material scientist would do, and thinking about it in terms of surface tension. So here, unfortunately, the contrast is not so good. It's due to optics. This actually is the same structure that you're observing here. So this is a structure with tissue being formed. In fact, this one is done before this one. This is the same scaffold here. If you can imagine the surface here, this black region is water. So this is during the washing process. And Cécile Bidon, who was working in the group and did the majority of these experiments that I'm describing, she noticed that, interestingly enough, the structure that you observe in the corners is quite similar to, when you have water in the pores, is similar to what you have in terms of tissue. So it suggests that maybe there's, say, surface tension could be of a tissue could be playing a role in the growth of material that you observe here. So you can model this, and this was work that we did together with Monika Rumpler, who's now in Vienna, who was doing a postdoc there. And if you take pores of the same surface area, so 2D simulations, and you allow the surface to move as a function of the local curvature, so Laplace law style growth, what you observe is quite a nice comparison between what you observe in experiments for different shaped pores, keeping in mind this is about 3 weeks later than these ones here. And interestingly, what you also can do is you can measure the amount of tissue from these very simple simulations as a function of time. And what I've plotted here is the area of these white pores as, well, the white area as a function of time. You notice that you take a circle, it's going to have the largest area for the same perimeter of all the other shapes. The slope of this curve is exactly the same as all the other ones. If I plot the green area, so the tissue area, as a function of time, you can't see anything. And why is this interesting? So in our experiments, we had this curious observation that if you look at square, triangular, circular, and I think hexagonal pores, one saw no difference in the global growth rate, the total amount of tissue that was formed in these pores. But we had observed local differences in the growth rate, because obviously you see, well, if I go back to this image here, there's more tissue forming here than on the flat surfaces. So this is quite encouraging. So we applied this to a variety of systems. where we looked at semicircular pores compared to round pores, and we run our simulations of curvature-controlled growth on these pores, and we can see a reasonably good agreement to experiments, at least at least for the first 15 to 20 days of culture. We see strange things occurring later on in culture, and this is something that we're not too sure about, but there's a few ideas about what's going on, and we can talk about that a little bit later. So what's quite interesting here is that geometry seems to be regulating at least the initial part of tissue growth. So playing with these shapes, you can come up with new types of shapes or architectures of these pores, and we can run our simulations on them. And interestingly, when you start making more complex or shapes, non-convex shapes, you see an acceleration of tissue growth would be predicted for, say, a cross shape compared to a square shape pore. We made these shapes, or Cecile made these shapes, I should say, and you see indeed in the experimental images more tissue formed on the right-hand side compared to the left-hand side, which you can quantify, and running the simulations also gives you quite a nice agreement with the experiments up to even maybe 20 days of growth. So this is quite encouraging. And you can quantify the difference in growth rates, and you can see that there is approximately a doubling in growth rate between the cross-shaped pores and the square-shaped pores, and shows that concavities at least promote tissue growth. So we're still talking about length scales which are much larger than the length scales I was talking about in terms of fibre orientation or fibril orientation. So we, together with Yves, Cecile, and Peter and myself, we started sort of thinking a little bit about what is actually going on, what can give rise to this sort of behaviour. And this is where, in fact, Cecile came up with this idea of this so-called chord model. And basically, if you can imagine a cell, it's quite a stretchy object. It can stick to a surface. When it sticks, it starts pulling. So there's this actomyosin contractility, which will enable cells to start pulling and feeling the mechanical environment. So if they're sitting on a convex surface, cells, when they start contracting, well, you end up with a resultant which is pointing downwards. If you look on a convex surface, then you would expect the resultant to be going in the opposite direction. So here one has a bit of a mechanism of how these tiny cells can feel these large geometries that we observed in the previous experiments. So this chord model is basically to say, okay, one approximates a cell or an object, a cell attached to a fibre or whatever, as a straight line. And when one starts placing that on a corner, for example, you see that you start filling in the corner, and interestingly, you fill it in at a rate which is proportional to the local curvature. So the growth patterns that one would observe based on this chord-like model, again, we're getting closer maybe to the real system, but we're still quite far off. It's still a geometric model. There's no rates or anything in here. The structures that one observes are at least comparable with what one observes in experiments. And if you look at the cell shape, excuse me for the resolution of this image here, you see that if you stain the cells for actin and their nuclei, you can see that they're very, very stretched in the corner of the pore. And if we allow tissue to grow even more, you can still see these highly elongated cells sitting in the tissue. So what's happening during tissue organization? So the process is that cells, or the idea is that cells contract, giving rise to a chord. So if you take a collection of cells, they develop tensile stresses, which gives you an organized cell network. Now, these cells are also producing fibronectin. So fibronectin, which we've stained here in red, you can see it's sitting underneath the main layer of the cells. And after producing fibronectin, they start producing collagen. So this is the main structural protein. And the collagen also co-aligns with the alignment of the cells. So this is quite interesting. So we see this spontaneous arrangement or organization of the cells, which then correlates well with the orientation of the collagen. So basically the model is that, well, first of all, these cells in green are sitting on a surface, followed by the fibronectin, and then followed by the collagen. Now, An important part of this is the cell contractility, so the mechanics of the cells. So how can one test that? Well, you can give drugs to inhibit the cell contractility, so blebbistatin, for example. And when you do that and you look at the cells, what you would expect, of course, is not too much to happen to the tissue. You would expect this tension to be released a little bit in the surface. And that's, well, maybe what you can see in this sort of experiment here. Of course, it's hard to quantify as you're looking at two different systems here. You can introduce an enzyme such as collagenase to degrade the collagen matrix. And if you do that, you would expect that this would result in a release of tension underneath that. And if you compare the control with the sample with collagenase, you can see after this collagenase treatment, you see this hole has decreased considerably in size, suggesting that a lot of mechanical tension is there. And the other thing, of course, you can do is you can come in with a laser microdissector, and you can now start cutting the tissue. And interestingly, if you do this, you observe this large elastic springback. And this springback is, on one hand, due to the tension developed by the cells, but on the other hand, it's also that if you do the same experiment but now inhibit how the cells are pulling on their environment, you see a residual elastic strain in the matrix. So somehow the cells manage to get the collagen matrix to be under a pre-stress, and that's sort of an interesting research topic to look at in the future. So perhaps to conclude on this section here, what I've shown you is that, so some preliminary work from our group of how we can explore how architectures are formed by living systems. The advantage of this system compared to, say, looking at plant tissues in a tree is here we can start really changing the machinery of how cells are creating and organizing the extracellular matrix. So to conclude, I think we've got about 15 minutes left for questions. I think what I've shown, or at least maybe put the seed in your mind, is that by looking at biological materials from a wider perspective, you can see that, well, many of the basic building blocks, one could say, are quite similar. So it suggests that architecture is quite important, which suggests in turn that this could be a rich way for material scientists to learn for designing new architectures. But one also has to keep in mind, this highlights an important point that I think was mentioned yesterday, is that for a lot of natural systems, they're multifunctional. So we don't necessarily know for what they're optimized for. And in addition, they're optimized under the constraints that us as mammals, we can't just decide, oh, I'm going to try having a bone made out of cellulose straight away. That requires huge changes in the biochemical machinery, which is not going to was not likely to happen at any stage. So that means that evolutionary constraints play an important role in the architectures that have been found by nature. I've described briefly how architecture controls function for plant tissues, how by controlling the microfibril angle or by controlling the shape of the cross-section of a cell, you can achieve many different functions. And then I've given you a little bit of an idea of how sort of processing by living systems can be investigated at least. And to highlight the importance of self-organization of these building blocks and the control of cells and the importance of mechanics on producing these sorts of systems. Of course, building man-made systems like this, we don't necessarily want to go in that direction, but the principles of self-organization, as was mentioned yesterday, these could be used for designing and building larger scale materials. So finally, as an outlook, so how could one then go and answer this question? I mean, this is something that's bugging me. As I mentioned, we can go for comparisons between different taxa, so looking at structure-function relations in very, very different systems, and to try and understand how biomaterials are processed. So what's happening during growth, morphogenesis, and coming with an engineering perspective, I think could be quite interesting to look at that. And there's a sort of a question here, which is sort of quite open, is would a CES-style database of structure and function in biomaterials be useful if one could then somehow couple that to phylogenetic tools, which are often used for, say, studying differences in biochemistry, etc. And this could be quite an interesting direction to go in. So with that, I would like to conclude and to thank, quite a lot of people here, so especially Peter, Mason, Cecile, Mickey, and of course Sebastian, Lorenzo, all the people in the group, and Yves, of course, for inviting me here, and Ingo, James, Pascal, and Ansgar for some of the images. And I thank you for your attention, and I look forward for some discussion over the next few minutes. Thank you.